I hope, what we can really focus on is presenting just some basic information for people in a calm way that doesn't involve sort of the vitriolic barbs that one sees on social media.
Do these prevent significant disease and morbidity and mortality in the human population? Because that's the long-term goal.
Welcome to Commune. We are on a mission to inspire, heal, pass down wisdom, and bring the world closer together. This is the Commune podcast, where each week we explore the ideas and practices that help us live this healthy, connected, and purpose-filled life.
So I'd love to just get into some basic virology before we attack the nature of the Delta variant. So if you don't mind just starting with the ancestral strain of SARS-CoV-2, so the original virus that seemed to emerge in China sometime in the fall of 2019, and really just explain to us how the SARS-CoV-2 virus infects a healthy cell, and maybe kind of in that explanation you can address a little bit of the much mythologized spike protein that has taken on a life of its own.
Sure. Yeah, I'd be happy to do that. Yeah. So, the first of all, the virus itself is a coronavirus, as most people understand at this point. And coronaviruses have been around forever. This is nothing new as far as our interaction with viruses as a whole. But this one is particularly novel. It has, various antigens and proteins on the surface that we haven't seen before. And therefore, the humans have no baseline immunity to this particular virus. And that is what's incredibly important here. Because viruses do what viruses do, but it's our lack of immunity that really creates a lot of the problem. And so what this particular virus does is, pretty simply, it has proteins, a specific protein on the surface, and we can talk more about the spike protein in a little bit and some of the mutations. But that spike protein specifically binds to a couple of different receptors on the surface of human cells. One of those receptors that a lot of people have heard about is the Ace2 receptor. And so once that binds, a series of biological steps happen that allow the outside of the viral membrane to then fuse to the cellular membrane, and then it inserts the contents. Once those two membranes fused, basically the guts of the virus then sort of get pushed into the human cells. Viruses are really, I like to call them beautifully stupid, and they are brilliantly stupid, however you want to say. But they actually have a simplicity that is quite elegant. But at the same time, these aren't organisms that think, even though you hear people almost giving them that ability. So all they do is they put their material into the cell, and their one job is to replicate. That's it. That's all they want to do. And they somewhat hijack our cellular machinery to be able to do this. So some of our machinery starts making new pieces of more virus, and then it brings along some of its own machinery as well, which allows it to then replicate in this specific case, its RNA. So all of that to say that really all it's doing is just trying to replicate. So once it replicates, it ends up, either leaving the cell or maybe even killing the cell. If there's so much virus that inside that human cell may actually die. So that creates a fair amount of the pathology. And this is not terribly uncommon with regards to how most viruses cause disease. So, so the virus is really not trying to do anything, majorly specific other than that. Once the virus has done this, it's repackaged and gets sent out of the cell. It can then infect another cell, and then the process just goes on and on and on until you have some element of an immune response that basically shuts that process down. Now, if you've ever had any sort of exposure to the virus or the proteins on the outside of the virus, then that immune response pops up pretty fast, and you're able to stop this maybe even before you even know that you were infected, before that you really have any significant disease at all. If you have no immune response, this virus gets to wreak havoc for a while until your immune system finally figures out what's going on. And by that time, there may be significant pathology that's happened, whether that be with this particular virus in the lungs, really in any organ system. People that have died from SARS-CoV-2 have pathology in almost every organ system that you can imagine: the brain, the liver, the lungs, the heart. It really is pretty impressive, mainly because it can cause problems with blood vessels. So that's it in a nutshell. That's generally all it does. It gets into the cell, it replicates, and then until it's checked, it keeps that process going until it ends up creating, pathology really throughout the entire body. It's specifically the lungs is where it starts and has its probably largest effect.
Nice. Thank you for that synopsis. So just to put that in a real life context, as I understand it, and please feel free to correct me everywhere and anywhere, is you're in a environment, generally an indoor environment, and someone coughs or sneezes or sings or expels some kind of droplet from their mouth, and to the degree that I understand, the virus spreads through airborne aerosol, essentially, and then you as a potential infected victim come in contact with that virus. The virus itself is encased, as you say, in some kind of lipid with these spike proteins on the outside. It comes down into your upper respiratory tract and connects to, as you say, the Ace2 receptors, then kind of fuses with the membrane of a healthy cell, penetrates the healthy cell, and essentially like hijacks its reproductive capability. Is that a fair understanding?
Yes, that is a fair summary. Absolutely.
Okay, so, and then it seems like the virus has a certain, I guess I might call it virality, or a certain amount of potency to spread. And once it infects someone, there is a reproduction rate, or I think what is referred to as an R0. So could you explain a little bit about what that means and how it was, and what the reproduction rate of the original ancestral strain was and is, and how that might compare to where we are now with Delta?
Right. So just a little bit about that particular value. It's an epidemiological term, and it really is just a value or a factor that allows us to comparatively look at the ability of a potential virus to infect from human to human. So, in a nutshell, even though it's sort of oversimplified, but in a nutshell, what that value means is how many people is one infected person expected to infect. So a virus that has an R0 of 2 an infected person would be expected to infect 2 others. And this could create, even at 2, this can create a pretty significant spread of the virus, because you can just essentially assume that there's going to be a doubling of infection over time. So that R0 value can be affected by preventative measures. So, the R naught value that we assign to a virus initially is usually assuming that everybody around is completely susceptible to infection and that you really haven't done anything to try to prevent it. So masking and non-pharmacologic measures can affect the R naught, and so can other things like vaccines and other preventative measures. So the ancestral strains of SARS-CoV-2 had an R naught of about between 2 and 2.5. And as far as viruses go, that's modest. I mean, it isn't. We've certainly seen other viruses that are more infectious than that, but it was something that was clearly causing significant problems, across Asia and then eventually across the world. But clearly Delta is a different beast that we'll talk about in a minute. Yeah.
Yeah. So, and I think failed out of math class more than once, if that's even possible. So, but my assumption or my presumption would be if you were able to bring a reproduction rate under 1, a virus would eventually peter out and go away, right? But as long as it's above 1, it's still in its spread phase. So we were dealing with an R naught of 2 to 2.5 with the ancestral strain. What are we dealing with in relationship to the 617 Delta variants? Yeah.
So one has to remember that it's been a little bit difficult to evaluate just simply because, number one, we have vaccines in the community, and number two, we're still gaining a lot of understanding of the virus. But it's somewhere between 5 to 8, which have been the estimates. The latest estimate I've seen is actually 6. Which means that one infected person would be expected to infect 6 other people. That's remarkable. It's not quite as infectious as chickenpox. It's certainly not as infectious as measles. Those are sort of the two examples that, classically within the field people give as sort of the uber infectious viruses. But it's in that ballpark, or close enough to where it's a very scary level of infectiousness.
Yeah, and so I've heard chickenpox can hover around a 10, and maybe measles even upwards of 16 or 18.
Right.
And to what does one attribute the higher R naught for the Delta variant? Why are we seeing a higher reproduction rate with this particular mutated strain?
Yeah, that's a great question, and it gets back to basic biology, right? So when we talked about the fact that all of these viruses like to do is replicate, in that replication process, it has to replicate its own nucleic acids. It brings its own machinery in to do that. And this is sort of where they're, sort of beautifully stupid in that they make mistakes. Their machinery makes mistakes, and it doesn't really mean to make mistakes. It just does. And this actually has an advantage for the virus. So in those mistakes, it sometimes will make spike proteins, for example, that are on the surface that have specific mutations. And so as time has gone on and this virus has been in the community, there have been thousands and thousands and actually millions of mistakes that have been made. And, there have been a certain set of those that have resulted in a variant. And we can talk more about natural selection and such if you like. But this is basically what has come out of that variation and selection process, is this particular strain that has a set of mutations in that spike protein. Now that spike protein, again, which is so incredibly important for binding the surface of the human cells, if it has modifications that make it stickier, that make it bind to the human cell more efficiently, then it's just naturally going to be more infectious. There's actually a beautiful study out of from Nature from late July, where the authors, have you seen this study?
Yeah,
I'm
I
not sure. Yeah,
saw it. Yeah,
it's
yeah,
really cool.
it is. Yeah.
So they where they talk about some of the the differences in the spike protein in the Delta strain as compared to the ancestral strains, and a lot of it has to do with just priming, if you will, those spike proteins, meaning that normally if you think of the virus and you think of the rendering of the virus, they always have a bunch of those spikes on the outside, right? Not all of those spikes are actually constitutively active, meaning they're not always ready to bind a human cell. In the Delta strain, 75% of them are ready to bind at any one particular time, and they bind really well. So that makes it really, really sticky to human cells. Where you might have, when I breathe, well, hopefully I'm not infected, but if someone breathes a million viral particles onto another person, and 999,000 of those just bounce off because they don't have spike protein that's ready, you may not get that much of an infection, or you may have a reduced ability to have infection. But if in the Delta strain, if half or more of those, 80% of those actually stick, then you're much more likely to have an infectious event. So that's the general concept, that these mutations have made it stickier.
Yeah, and I think I read an analogy that you made where the ancestral strain might have had a spike protein that could be compared to like Elmer's glue,
Or syrup.
or
Yeah,
syrup, right?
yeah, yeah,
But
yeah.
the Delta strain seems to more resemble your industrial gorilla glue, right?
Right.
Which I thought was helpful to explain. And then can you make the logical leap between this enhanced, more potent spike protein, or more potent binding mechanism, and viral load, which seems to be much increased in folks that are infected with the Delta variant?
Yeah, we're studying that as well, and so there are two hypotheses that one could make here. Number one, by the time one becomes symptomatic with a particular virus, there's a certain amount of viral replication that has happened. And so we're either this is in the back of the throat, right? So this is where we actually detect the virus. So the back of the throat or in the nose, right? Either one.
Yeah.
So there's a certain amount of replication that's happened. If you start at a much higher level, because so much more virus has bound to start with, you're just going to have more virus at the point of when someone becomes symptomatic, as compared to the ancestral strains where you may have just started with, say, 10 or 15 viruses. Now you're starting with 10 million, and it's just a numbers game. And right. So the other possibility, and that we're actually looking at, and there is some evidence for this, is that the Delta strain actually does replicate faster. That it does have some advantages with regards to its ability to reproduce inside cells in just a little bit more efficient way. So we're figuring that out now.
Yeah, it seems like I've also read about these kind of infection clusters that develop that seem to be more pervasive and insidious. So can you then talk about the greater susceptibility of young people, and I guess of cohorts that seemed to be more unaffected with the ancestral strain? When SARS-CoV-2 really came on the scene in force, it seemed like it was more aggressively impacting the obviously elderly and the immunocompromised. But with the Delta, that there's that Venn diagram seems to have expanded a little bit more. So I wonder if you could just address that for a moment.
Sure, and this just gets back to basic microbiology, infectious disease, and I think I'd written something very kind of folksy about this before, but if and indeed it's true, if you spew enough of a human pathogen on someone who's susceptible, there is a good likelihood that they're going to get a significant infection. And we think that's exactly what's going on here. That I mentioned a numbers game before. That's really what this is. That before and younger, healthier people, there may have been enough of an innate immune system response that's not specific to the virus. It's just an innate system we all have, sort of at the surface, that was able to ward off the small amounts of virus that one might get casually during certain infectious events. Those innate systems aren't that great and as you age, et cetera. But now, because you're being bombarded with so much viral load in any one infectious event, that innate immune system is really rendered pointless. It just doesn't. It can't even come close to touching it, and so it's causing infections in younger populations. The other part of this is our younger populations tend to be our unvaccinated or less vaccinated population right now. Our older population rolled up their sleeve really early and were actually vaccinated and thus have protection. We can talk about breakthrough infections later, but yeah, that's generally why we're thinking that this is now a pandemic that's demographically spread out over the age groups.
Yeah, I have 3 daughters that are headed back to school over the next week or 10 days. So the timing here is certainly a source of some concern. Obviously, we want to get our kids back to school, even just for our own sanity, but just for their ability to learn and to socialize, et cetera. But it's coming at a time that is pretty fraught. Maybe we could just spend just a moment just on evolutionary biology to understand what are the pressures that are creating variants, and I think it should be noted that there are plenty of variants that seem to be not particularly potent at all. But obviously, in the case of Delta, that's an exception. So, and there have been a lot of theories posited around this. I mean, I've even read some theories about actually vaccines creating some pressures that would have sort of the virus naturally select for a variant of higher virulence, if you will. But those seem to be somewhat on the edge. But yeah, I wonder if you could just kind of give us a little primer on how variants actually develop.
Right. So this gets back to the point that the viruses really are simple, and they really have one goal in life, and that is to transmit. It's to transmit and replicate. And so when you look at, and let's get back to variation as well. Variation, as we discussed earlier, because of those mistakes and that replication machinery, variation is always going to happen. And indeed, I oversee a variant sequencing lab here, and so we're watching this in real time. And you are correct. There are gobs and gobs of variants that have been detected to this point, and most of them nobody cares about. You've never heard of them, nor do you care to hear about them. Yeah. They all have a string of letters that we've denoted them by. You just don't care. So, we care because we want to make sure that it's not something the next wave. But if you think about this big, massive pot of variants that come out of this, that all have some little change or the other, why does one pop out of this and create major disease issues where the other ones just sort of fade away into nothing or never turn into anything. And it all has to do with which one of these variants can transmit better. Because again, that's all the virus, that's all that makes a particular variant successful, is transmission. So what gets in the way of the virus transmitting? And, to this point, the only thing that's gotten in the way of the virus transmitting are just general natural factors that have existed. They could be elements of the innate immune response that we talked about earlier. They could be elements of, its binding to human cells. It could be its ability to aerosolize a little bit better. Those sorts of things. But almost all of the variants that have been successful have had mutations in that spike protein. So they've all been, just had a better ability to bind to human cells. So the concept that a vaccine will actually drive this level of variation is both true and false, right? So the vaccine itself is indeed something that gets in the way of viral transmission. But what's really pivotal and very important to realize about the vaccine is that it's not easy to get around because of the way it's designed, because of what we're actually doing with the vaccine. It's going to be highly unlikely that any particular viral variant is going to be able to completely get around the immunity that has been generated by the vaccine. Mm-hmm. Will we have variants that have somewhat blunted, maybe reduced, or maybe we have reduced immunity against those variants? That is quite possible. And in fact, that may be happening somewhat with Delta, potentially. But to say that the vaccine actually caused that is actually fallacious. It's actually not true.
Yeah, I mean, obviously, all of these strains that we're talking about now were precursors to the rollout of the vaccine. So just even from a logical standpoint,
Delta arose in India where there was, essentially no vaccination at the time. Right. So
yeah, absolutely. Right. So now we're looking at a mutated strain with a reproduction rate of approximately 2 to 3x the original. And there is an emerging rate of breakthrough cases that seems to be fueling concerns that the vaccines are less effective. And we can go into whether or not that's true or what the reasons are for that, if it is true. But I think just to step back, in order to understand the current effectiveness of the mRNA vaccine, specifically the Pfizer and Moderna, maybe we can do just a little vaccinology 101. Mm-hmm. So can you explain how the messenger RNA vaccines were developed and their basic functionality?
Yeah, so there are really two aspects of this. I think to fully understand it, we need to understand a little bit about how we actually vaccinate against infectious diseases, the classic ways that we do this, and then how the mRNA vaccine works. And I wrote a fairly lengthy Facebook post about this not that long ago. But classically, if you want to immunize someone against a particular pathogen, you could either take some live or attenuated pathogen or even killed pathogen and put it in a buffer and inject it into someone. And all of the various proteins and such on the surface of those things would stimulate an immune response. You would then, now have that on board, and you'd be able to react to any future, live infections with that bug. The other way that you could do it is just to make in the lab, make those proteins, not the whole virus. You just have those proteins, put it in a buffer, inject it, and stimulate that same immune response to those proteins. And again, that classic way works really well. It's just really, really slow. It takes a long time to be able to produce those proteins.
Yeah, that was actually one of the questions I was going to ask you, is why there hasn't been the typical, I think they called immunogen vaccines, kind of like the Salk polio, where there's an inactivated virus. But is the principal reason why we haven't seen those vaccines just time horizon for development?
So as far as the killed vaccines or the live attenuated vaccines, yes, those take a while to actually safely produce. If you're talking about historically the types of vaccines that generate significant side effects, it's that class of vaccines. Just because they're dirty, they're messy, they're just a lot of different antigens in there that actually can lead to Guillain-Barré, which is a neurologic side effect. So so that's been the main reason. Now, as far as the protein-based vaccines, which are much safer, where we generate the protein and put it in the sort of mix of buffers, those are actually in trials. There's a big trial in the United States on one right now. And there are some that have been used across the planet. The advantage to those is they're very easy to distribute. And you can make pots of it very quickly. But yeah, it's that has been time has really been the major issue with that.
Yeah, and obviously because the messenger RNA vaccines appear to be a new technology. Though my understanding is that they've been researched for some time, it has caused some uneasiness. Just because the period of development was so quick, and that there's never been a licensed or even emergency use authorized vaccine that leverages this particular technology. So that's created a lot of public uneasiness in accepting these. But, maybe this is a good bridge into that technology and your understanding of how that technology functions.
Sure. So, and again, a little basic biology primer, and then we'll talk about how they're produced and actually what's in them. So, we all have, in our cells, we all have DNA, and that DNA is the blueprint for all the many thousands of proteins that your cell needs to make. You don't want your DNA actually just hanging outside that nucleus at any point in time if you can avoid it, because the cell would die. And so, but that message that the DNA holds needs to somehow make it out to the protein machinery, where the proteins are made. And the bridge for that message is a molecule called mRNA. When a protein that is needed to be made by the cell, there is a process called transcription, where mRNA is made off of the DNA template. It then goes to the cytoplasm, to the protein production machinery. That mRNA is then read by the protein production machinery. The protein is produced. The mRNA is then rapidly degraded. If there are any biologists out there that have ever worked with mRNA or worked with RNA period, you know that RNA is incredibly unstable. It's very susceptible to lysis and physical degradation, and that's by design, because you don't want the RNA to just keep making protein. That's not how it's regulated. So that's the natural biology. So the idea here is that instead of making all of that protein from a particular pathogen in the laboratory, can we just give cells the mRNA template for that particular viral protein and let our cells make the protein for us? So it's the same natural protein production process that your cell is using right now, all of your cells right now, hundreds of thousands of times, in order to make protein. And so we put that mRNA into the cells, and we can talk a little bit more about the very gentle process where that has to happen. But the mRNA goes into the cells. It's read. Protein is made. In this specific case, it's the spike protein that's made from the virus that's made, and that protein is then released. It's considered foreign protein, just like it would be if it were injected just a straight protein in a traditional vaccine. The immune system sees it, creates an immune response, and then off you go. You're now immunized or beginning to be immunized. You're training your immune system to react against that protein. So that mRNA, in order to get it into the cells, it's very simple. And this is where the technology it's been under investigation for at least 25 years. I had a buddy of mine remind me that when we were in graduate school back in the mid 90s, we had a really interesting talk where we were at school at the University of Arkansas from a University of Texas Southwestern investigator that was talking about this very specific thing. I think this was in 1996. So this has been this idea and these types of things have been going on for a long time. But in order to get that mRNA into the cells, it's literally just packed in some lipid. It's very simple. There's really nothing to it. There are no stimulants or anything. And that lipid is just to keep it stabilized and to allow it to basically fuse with those human membranes in order to safely get the mRNA into the cytoplasm. Then along with a little sugar and salt, that's the mRNA vaccine. That's it.
Got it. So let me retrace a few of those points. So we were talking earlier about the spike protein and how this is the connective element that provides the envelope through which the virus attaches and infects your healthy cells.
Correct.
So that became sort of a target for scientists and vaccinologists early in this process. They're like, okay, if we can attack that spike protein, then we can essentially neuter or the virus's ability to enter the cell, infect it, and hijack its reproductive processes. So that's what they did. So they mapped the genome of that spike protein or of the virus as well, and they basically created the message that the DNA would normally send out into the cytoplasm to the ribosomes, and they essentially made that in a lab, and they packed it in some lipids, a very simple ingredient list, as far as I understand. It doesn't have any adjuvants or anything like that.
Correct.
And then this is injected into the muscle in your arm, and this mRNA then essentially goes into your cytoplasm, messages a protein production process to produce that spike protein. Your body recognizes that as a foreign agent and gears up your adaptive immune system, and then you start to create antibodies for that. Is that a relatively fair understanding?
That's a very good summary. Yes.
Okay, so one question here, and this is getting a little into the weeds, but there has been some theories promulgated that the spike protein that is associated with the virus has some detrimental impacts separate from the virus itself. That when it attaches to the Ace2 receptors, that it can damage mitochondria, et cetera, and contribute to some detrimental health impacts. Now, I don't know if that is your understanding as well, but if it is, what I have read, and maybe you can clarify this, is that the spike protein that is generated through the mRNA vaccine is slightly different in a couple amino acids, and this is cutting with a very fine blade here.
Yeah, talking proline substitutions, right?
Yeah, exactly. Such that the spike proteins that are created by the vaccine don't seem to have the same deleterious impacts than the spike proteins that are concomitant with the virus itself. Could you unpack that for a moment?
Yeah, so that's a good summary, Jeff. And anytime you're actually creating vaccines, you want to make sure that the protein itself is not pathogenic in and of itself. It's very uncommon that it is, mainly because proteins that sit outside the context of a pathogen, usually, unless they're true toxins, they usually and viruses don't classically make toxins. They make a few, but this is not necessarily a toxin per se. But you want to make sure that the protein itself isn't itself pathogenic. And so there were a couple of reasons for making those amino acid substitutions. One is that those substitutions actually opened up the protein, and the reason you wanted to do that, if it folds on itself and you can't, and your immune system wouldn't see all of the important pieces of it in order to make all of the correct neutralizing antibodies. So that's one goal, and the other is to also render it essentially non-effective with regards to binding some of the important aspects of other biological receptors, et cetera. So and again, if the protein itself that's produced is not creating, and we should talk about side effects of vaccines. Yeah, because it kind of relates to that. I mean, that's what this really gets to, is that, are these vaccines, safe? And it has to be, one has to remember that we've given millions and millions of doses of these vaccines to this point, and the side effect profile is very, very small. Anytime you develop a drug, you're going to have a certain number of side effects, especially when applied over a large population. And the average, the average side effect profile of any particular drug is going to be what it is. But this is extraordinarily small. There's some myocarditis and pericarditis in a small number of people. It's usually teenagers and over the age of 16, and they're usually male. And it's usually after the second dose. It's really narrowed it down. It isn't very common at all. So did you have something?
Yeah. I mean, I think I've seen a statistic that it's about 12 cases per million. Per million second doses actually administered, to be specific. So the side effect profile seems to be limited there. And I think I think there's a lot of loose verbiage that gets thrown around with regards to the vaccine. It's in many cases called experimental, even though it did get FDA license this week. But perhaps you could just very briefly address some of the initial efficacy data and the trial process that these vaccines underwent.
Sure. Yeah. And I want to begin by saying that every drug is experimental when it first comes out, right? Even after FDA approval, we always watch drugs very closely, even after they hit the market. But this particular vaccine had a very unique, both of them, both Moderna and Pfizer and J&J, all three of them actually had a very unique pathway to approval. What was first emergency use authorization, and now with the Pfizer product for full FDA approval. Classically, when you do trials for a particular drug, you are going to do them in sequence. There's phase 1, phase 2, phase 3. You have to determine whether or not the drug works. You have to determine whether or not the drug's safe. And then you want to replicate that over a much larger group of people in a phase 3 trial. And, if you think of this from the point of view of a drug company, those trials are incredibly expensive, very, very expensive. And they actually bring a lot of drugs into trials that fail. And so you wouldn't want to do a phase 1, a phase 2, and a phase 3 at the same time and spend all of the money for all three of those phases if you aren't really sure that the drug is going to work, number one. So you would almost never do that. So normally you would never go to phase 2 without making sure phase 1 works, never go to phase 3 without making sure phase 2 works. Here, because we needed them so quickly, there was enough money on the table, if you will, to allow them to do all three phases at the same time. And so we actually generated the same amount of data that we would in a typical, or close to the same amount of data that we would in a typical trial, sequenced trial period. Not only that, there were more people looking at this data than probably any other drug class in the history of any drug. And so there's so many people so many scientists were looking at this because we were all very interested. We were all also about to get injected ourselves, right? So we wanted just self-preservation, wanted to make sure that we weren't doing something that was going to be deleterious to our own health. So because of that, it was allowed. It was able to come to a market very quickly, very much quicker than your typical pharmaceutical. Not only that, I also want to point out that something you mentioned earlier on. We were able, because of our history with coronaviruses, specifically SARS-1. We were able to, based on experience from that, sequence SARS-CoV-2, get the genomic sequence from SARS-CoV-2 very fast. So we knew the genomic sequence very early. And then because these mRNA vaccines are so easy to produce and so quick to produce, and because we had all of this information in January of 2020, basically, we didn't have any of the lead-in time that you would typically have for a vaccine. So not only did we have something to test very soon, we were able to take the trials and stack them on top of each other and get a product out to the public in a very short period of time. It doesn't mean we short-circuited the whole process. We just did it in an intelligent way with a lot of brute force work by a lot of scientists.
Yeah. And maybe you could take a moment and explain what the nature of randomized double-blind trials look like. So, for example, with Pfizer, I believe in the third stage clinical trial in the fall of 2020, there was about 44,000 people in the overall trial. I think in Moderna, closer to 20,000, maybe just north of 20,000. So how are the vaccines and the placebos administered in a way that reveals effective data?
Yeah, of course. And any double-blind trial like this means that the person who's receiving the vaccine and the person that's giving it have no idea, what they're giving. If they're giving, nothing but just saline, or if they're giving the actual drug. They look the same. They're labeled the same. You cannot tell the difference. The only place where that's known is on the back end, because each one of those has a barcode on it, and so it has a specific number. So in the background, someone knows. So what's interesting about these two vaccines is that they were just so remarkably effective as compared to placebo in those trials. And same way with the safety data. It was also, again, even in the 44,000 and I think 21,000 or 22,000 in the Moderna.
Right. So I think if I understand it correctly, the placebo and the vaccine were administered, and then there needs to be enough virus generally out there for the trial to be effective.
Correct.
And so they're constantly testing these groups, and I think they test until 100 people test positive, and then they kind of pull back the curtain and they say, well, of these 100 people that tested positive for COVID, how many of them had gotten the vaccine and how many got the placebo? And it was something, to the tune of, 95 of the 100 had been unvaccinated and 5 had been vaccinated. So there was an infection component of the data, but I think, and this is what I'd love to get your opinion here, it seemed like the really compelling component of the data, though, was that of the vaccinated group, virtually nobody was hospitalized. I mean, I think maybe one person had an oxygen level that was lower or something, but to me, and I think if you're going to put the vaccine efficacy on a hierarchy, it seemed that what the shining light of these vaccines is that it seemed to prohibit aggressive contraction, essentially hospitalization and fatality. Is that fair?
Yeah, and I think that's a very important discussion to have when you're talking about effectiveness of vaccines, like effectiveness toward what endpoint. There's an effectiveness towards the endpoint of just infection itself. And then there's the endpoints that most vaccines really have, which is, do these prevent significant disease and morbidity and mortality in the human population? Because that's the long-term goal. Now, of course, we would love for it to prevent infectiousness. We would love for it to do that. But the real end point is, we want to make sure that the vaccines are preventing severe illness and death. And you are correct that the vaccines in that sense were not only remarkably impressive early on, they've remained that way, and they still remain that way, even through the Delta surge to this point. For that particular aspect, they have remained very effective with regards to preventing hospitalizations and death.
Yeah, and so just chronologically moving forward, the third stage trials happened. The data came out in November or so, 2020. There was emergency use authorization granted by the FDA, I believe, in December 2020. Mm-hmm. And then concurrently with that, we were seeing huge spikes starting in January in deaths. I believe we were somewhere, north of 4,000, even a couple of days north of 5,000 per day in January. And then the vaccines began to be administered and started to permeate, through the various cohorts. And there was a significant decrease both in cases and certainly in fatalities. But so at the same time that we were seeing that good news in the United States, many of us were watching what was happening in India, and the Delta variant began ravaging India, and particularly seemed to be impacting younger people. I mean, India is a little different demographically. I think the average age there is 28, so it's a little different case study. But eventually that variant inevitably came to the United States, and I believe it's now the dominant strain. And now we've seen a spike in cases and deaths, which always seem to lag a little bit behind. Mm-hmm. And more vaccinated people in the U.S. are starting to test positive, including my brother 2 days ago. So maybe you can address the issue of effectiveness of the vaccines vis-a-vis Delta, and if they are less effective, then why is that exactly?
Right. So again, some of this is hypothesizing at this point, because we're still learning, and again, there are important points for us to be able to define, because they really do outline our next steps forward. And so the question is, first of all, the vaccines are still, as I said, remarkably good at preventing hospitalizations and death. If you look at the areas in the U.S. that have had just remarkably high levels of cases, there have not been a significant number of hospitalizations and deaths in the vaccinated population anywhere near compared to the unvaccinated, but they're still happening. And a lot of the patients who are vaccinated and become infected and get significant disease and even die are typically have some other comorbidities, some other disease process, or maybe are elderly. Maybe they didn't react to the vaccine all that well to start with, or some element of immunocompromise. But still, there are some breakthrough infections that as you said, even in the younger, healthier populations. So the question is this because this viral strain is just incredibly good at what it does? And I think that's a yes. It is incredibly good at what it does. Or are we actually seeing waning immunity? Because most of us, or a lot of us, were vaccinated in January, February, March, and we're just starting to see, especially amongst the people that were vaccinated early, are we seeing waning immunity that's leading to this too? And there is some anecdotal evidence to suggest that may also be true. So we may have two things converging here at once that are leading to some of these breakthrough infections. This is exactly the data on the latter piece is exactly why, the White House and the CDC are strongly considering, booster doses sometime early next month. Is to try to do this. But again, this is to prevent infections and possible, less, try to prevent infections, but more than anything, to try to prevent some of the more severe disease and maybe some of the few people who are actually debilitated and may be elderly that didn't react so well the first time around.
Yeah, and do you think we'll see eventually boosters that are optimized for the Delta strain? Because obviously these vaccines were designed for the spike protein back of January 10th, 2020.
Right. I don't think that I'm going to try to be clear here. I don't think that the immunity that is generated secondary to the vaccines on the market now are a problem with regards to Delta. I think they're still very effective against Delta. Delta just creates so much viral load that comes at anyone that even if you had a vaccine that was exactly the same spike protein, you may still be seeing this exact same phenomenon. It may have nothing to do with that. So just simply because there is good evidence that we're still mounting a very strong immune response, against this particular virus.
Yeah, maybe you could talk for a moment just about the incubation period and where the antibodies seem to exist within us physiologically,
right.
Because I think that's actually a key point, and you can get into, I think, some of the differences of hemoglobin and stuff that's beyond my pay grade.
IgA and IgG, So we when you vaccinate or someone and what you're generally going to get, when that protein is released from those muscle cells, is you're going to get that amnestic immune response that's going to generate immunoglobulin G or IgG. And that's the immunoglobulin that floats around in your blood. You can find it in some of your tissues, but it's it's sort of the circulating henchman of the immune system, if you will. Over time, it will, if it doesn't see enough of that protein, it will sort of wane, and you may not have nearly as much of it floating around. But at the level of the throat mucosa, the nasopharyngeal mucosa, IgG, that's just not where it hangs out. That's not the kind of antibody that you usually see on mucosal surfaces. This is the mucosa there, the mucosa of your gut. Typically, mucosal antibody is IgA or immunoglobulin A. The vaccine, while it produces some systemic IgA, does not necessarily produce IgA at the level of the mucosa. And so when you're exposed to a particular virus for which you might have good IgG related immunity from the vaccine, it doesn't necessarily mean the virus can't still attach and replicate some. And because of this particular variant, and because there's so much of it there, it can actually get a little ways down the infection process before the IgG actually gets involved and says, we're not having any of this, right? So you may get transient infections, and you might even have someone who gets, kind of mildly symptomatic. If the IgG levels have waned to the point to where they're quite low, then you might even be able to have a fairly significant infection. Or if you never had IgG levels that got all that high, from the first rounds of vaccination, then you may be able to have, may end up with having a pretty significant infection. And, this happens over a course of a few days, anywhere from 2 to 5 to 6 days. And indeed, what we've seen is that in persons who are vaccinated and get infected, that the levels of virus in their throat are, at least for the first 5 to 6 days after they've been infected, are the same as someone who's been unvaccinated. But at that 5 to 6 day mark, the virus in the throats of the vaccinated person starts to drop remarkably, and that in the unvaccinated person remains. So so it's
so this would also affect potentially the rate of transmissibility between a vaccinated cohort and an unvaccinated cohort. And what I'm sort of honing in on slowly is that there are plenty of people right now that are saying, well, there's breakthrough infections, and it seems that even vaccinated people can transmit the virus. So why would I get vaccinated? And I think you've addressed a number of reasons, but maybe you could just bullet point some of those.
Yeah, I will start with that one because at day 4 or 5, in the people who are infected, right? So we can talk about the 3 to 4-folded reduction in the infection rates in those that have been vaccinated versus unvaccinated. But in those that get infected, you're only infectious for maybe 5-6 days. Maybe 4 to 5 days, where someone who's unvaccinated can be infectious for as many as 18, right? So if you're thinking about this from a community spread standpoint, the vaccinated person is certainly not a vector for nearly as long as compared to someone who's unvaccinated. And the other part of this is just the protection of your own self. Again, the severe disease, the COVID long hauling, all of the things that are, that this virus has led to with regards to morbidity, and then also the mortality, because we are seeing deaths even in younger people, young, healthy people, it's really just a protection. We're not seeing those in the vaccinated population. We are seeing some deaths in the vaccinated population, but those deaths are, as I mentioned, are primarily in the elderly population and folks that probably did not amount to a very good immune response to begin with. So the main reasons that one would want to get vaccinated at this point would be to, number one, protect themselves, and then number two, to protect others. Just simply because while you still could be a vector, you're not going to be a vector for nearly as long. And because you could still be a vector, this is why the CDC and others have said, okay, we've got to go back to the non-pharmacologic interventions. It's not something we really want to do, but we need to go back to doing the masks and distancing as much as we can tolerate as a society. You'd mentioned in the front that having our kids in school and the importance of that, and I think that's fully understood. And so, but there are still common sense things I think that we as a society can agree to do to try to protect one another. And some of that is getting vaccinated, and then also doing the, sort of the common sense, non-pharmacological things that not only protect you, but protect others.
Yeah. Are there any therapeutics or prophylactic therapeutics that you advocate for either in conjunction with vaccination or instead of vaccination?
Yeah, it's not really my area of expertise. I'm not an infectious diseases treatment expert, and I know where you're going with this, because so it's probably either So I've had a lot of questions about this, about this particular topic, and I kind of talk around it. So but I will say that there are monoclonal therapies that have been, very heavily promoted as being, some sort of ability, or give us the ability to potentially get around this. Well, first I will say that the delivery of those pharmaceuticals is not simple. And operationally, trying to actually deliver those above, or deliver those instead of delivering vaccine is much, much more difficult, and it's much more expensive. So it's sort of an ounce of prevention, pound of cure type of thing. Now, in a very heavily infected population, if the horse is already out of the barn, meaning that you're in an area where there are so many infections and that it's almost too late for a vaccination campaign. You may think about certain areas in Florida, maybe, certain areas in the southern part of the U.S., Alabama, maybe Mississippi. If you could provide that on a very controlled basis to try to reduce the amount of hospitalization, then that could be a truly helpful public health measure, just simply because it keeps our hospitals functioning and not completely full of COVID patients. So if it has that effect, that's great. But as a general public health overarching strategy to try to get us past this is just not terribly wise. It's not efficient. It's certainly not economical. And there have been some other drugs that have been mentioned as people think that they're they're the old wonder drug, that's the new wonder drug, and there's just no data to actually suggest that they have the effect that people think that they do. And multiple societies and agencies have strongly urged against their use for many reasons. So we're sort of back to the hydroxychloroquine arguments again, unfortunately.
Yeah, and ivermectin and fluvoxamine and
a bunch of ivermectin's the new hydroxychloroquine, right?
So as people kind of assess the risk benefit, or they perform that analysis, one of the factors that they're weighing is like, I can take my chance with Delta, or I can go down to CVS and actually, get an injection, which seems like it's a more proactive choice. And in that proaction, you are having to address risk benefit. So I know we talked a little bit about side effects in myocarditis and pericarditis. What is your assessment of some of the reports that have appeared on VAERS, the Vaccine Adverse Event Reporting System, around COVID vaccine-related deaths or other kinds of side effects, and you might have to provide a little bit of context on VAERS itself.
Right. Yeah, I think we'll start there, actually. So, anytime that you're trying to gather data like this, population level data, you have to cast a very, very broad net. And so you're actually going to have very high sensitivity at the expense of specificity. And that's exactly what VAERS is designed to do. It has almost no controls on the front end, meaning anybody. I could go in right now and say, yeah, I actually have a broken toe and I think that it's related to the vaccine. I mean, you can kind of manipulate it if you want to. You don't even have to be all that, truthful or honest. It's the hope that people are. On the back end, there are various tools that are used by epidemiologists and scientists to be able to wade through that gigantic pile of data and try to come up with, are there truly any links between all of this gigantic data set and actual side effects related to the vaccine. What's been, unfortunately, what people have done is they've taken the front end data set that is very noisy, that is very full of just non-contributory information, and turn that into, actually said this is fact. This is exactly how we do this. I do the same thing as well with when I have to report when we do blood transfusions. I'm on the transfusion utilization committee. And, any death that happens within 72 hours of a blood transfusion has to be reported to the FDA. And we have to actually, or we at least have to review it and determine whether or not it needs to be reported to the FDA as a contributing cause to the patient's death. We do this on a pretty common basis, these reviews, because, as you might guess, there are a number of people that, a lot of people that get blood transfusions are fairly sick, and so there are deaths in this group of patients. And but when we go back to look to see if there was any causal link at all, almost never. It's very, very rare that the blood transfusion itself had any sort of causal effect. Theirs is the same sort of data collection process. Again, you cast the wide net, then you have to go in and determine whether or not there is a causal link. So, from a population standpoint, when you look at the data that has been reported through that system that has been accurately evaluated, and the data from the trials themselves, there just is no at this point, we're at a point to where we can make a really good systematic evaluation of safety. There's no significant side effects associated with this vaccine that happen with any sort of frequency. I mean, many of these are just extraordinarily rare, as you said, 10 to 12 in a million, and that may be a slight overestimate. So,
yeah, nice. And I suppose, what VAERS really does, it's an early warning system for professional data crunchers to assess,
right.
And it seems to be doing its job, sort of with regards to some of the blood clots that were associated with the low platelets tied to some of the AstraZeneca,
the J&J, and AstraZeneca, yeah.
And because that event is so very, very specific, it makes the attribution of causation easier. So it seems to be effective there. There are these like 12,000 fatality reports on VAERS, and of course there's no causal link there at all. But even if one accepted those on face value, if you were doing any mathematical calculations, you would find a fatality rate associated with COVID, 100x times the fatality rate there. So I just,
right.
I try to underscore that as much as I can. Yeah.
Right. Yeah. No, I agree with that. But I still think when I talk to people, I still try to make sure that they understand that 12,000 number is much like our transfusion number. That's an upfront number that has really no meaning whatsoever. It's the number on the back end after the evaluations that have been done that are so incredibly critical to look at, and those numbers are essentially zero. We don't see people coming into our hospital on any kind of regular basis, secondary to any kind of vaccine or vaccine side effects. It just doesn't happen.
Yeah, yeah. So, just in closing here, of course, this is time stamped, so we don't really have to worry if it ages well or not. But I'm going to ask for some. We've done a lot of diagnosis. I'll ask for a little prognosis. Where are we headed in the short term and the longer term? I mean, are we headed towards endemicity, or are we?
Yes.
Should we expect or fear a variant worse than Delta, or where are we?
So where we are along the way, and again, I've written that this is in a way this is kind of a choose your own adventure book. I do think that the end itself is kind of written in a way that it likely will be endemic. So we'll start there. We'll start at the end point. This is very likely to be something akin to flu. It possibly could be reduced to something no more than cold-like, a cold-like illness, just simple upper respiratory illnesses. But it may actually be more like influenza. The good thing is that this virus actually doesn't vary nearly as fast as influenza. It seems like it's super fast, right? But that's just because there's so much replication happening in the community. But once we get on top of it, we may not actually have to booster this thing more than once every couple of years, or maybe it would just be a booster that would be combined with the influenza vaccine each year, maybe in a single mRNA vaccine that covers both of them. And so there is an endpoint here to where this just becomes part of our background. So how do we get there is really the question, and that's the choose your own adventure part. The least painful way is to try to get as many people vaccinated as possible, so that we have a reduced level of disease and death, while we actually get and generate enough immunity in the community to reduce spread to the level to where we can just make it an endemic background virus. And indeed, amongst the vaccinated population, that's kind of where we are, at this point, because what's going on right now is within the vaccinated population, if you just completely separate them out, it is kind of what it would look like in an endemic scenario. Maybe a little worse. I mean, we may be able to make it a little bit better just with boosters and more longer term immunity, better trained immunity. But it's really the spread and the effect of on our hospitals and such in the meantime that are really painful. This fall is probably going to be the next 6 to 8 weeks is probably going to be really painful. In a lot of areas of the country. I think there's almost no way around it, unless everybody went and got vaccinated tomorrow. I just continue to try to, talk to folks and try to educate them as much as I can, and try to get them and to, just understand the situation and that they do have an option here. They can choose their own pathway here and protect themselves with the vaccine. I understand that it's somewhat scary in a way because it's a newer technology and such, but it's not that new now. It's been around for it's been around for 25 years, but it's been on the market now for over a year. Or at least in trials for over a year. Just really, really wanting people to think about this, this risk, as you mentioned, in the whole, and make decisions that, are the best for them, based on everything that we know now. But I also understand that there's so much information out there coming from so many different places. And then if you don't have a background like mine or people who are, who read a lot of science and understand it at its baseline, they would be really, really difficult decisions to make. And again, I've gotten questions from people all over the country, all over the world, actually, that just demonstrate how confused and how really unsure and hesitant that people are, not because that they have anything against the vaccine. They just really don't understand a lot of the basics. And so and that's just sort of been my goal, is to try to shed a little bit of light on that, and just arm people to make the decision, or better arm them to make decisions.
Yeah, well, you do that so well, and I know I'm not alone in saying that we're grateful for your work at this time, and not just for the work you're doing, but how you're messaging around the work. You're obviously passionate about what you do, but you express it in a way that is aptly dispassionate, if you know what I mean. Such that there's so much shaming going on in every direction, and the ecosystem on social media can be so vitriolic. And so I think the path that you've chosen, which is just educating people on the baseline, is the best possible path, and I really applaud your efforts. So thank you so much for what you're doing, and for spending time with me today. I appreciate that. Hey, thanks for watching. If you liked this interview from the Commune podcast, then I think you'll love this video right here. And if you want to get more weekly content from Commune, you can subscribe right here on our YouTube channel, or find the Commune podcast on Apple, Spotify, or wherever you get your podcasts. I'm Jeff Krasno, and I am here for you.